எனக்கு PDF வேணும், பக்கத்துல ஒரு எம்டி ஸ்லைடு வேணும். Okay, sir. Okay. Okay, sir. Shall we start? You have your yellow material? No. But anyway, you have it with you, right? Fine. Okay. So when you not a chat, initially it was seen, you can't see. Okay, fine. So now, see Hari, and please go through like the textbook, okay, for the previous things which discussed. Okay, I think it is easy for you to follow, and directly we will come to the Mendel's experiments. Okay, just go through this paragraph. It says, listen to me what it is there. Okay, I'll tell you. Graham Mendel selected 7 contrasting characters from the pea plant. First point. What is the first point you should know? Graham Mendel selected pea plant and actually 7 contrasting characters. You know what are the 7 contrasting characters? Okay, in your yellow book it is there. Can you tell me? Look at here, Hari. Pea plants, round or wrinkle seed, tall or short plants, violet or white flowers. Can you see? Like that, there are 7 characters. Okay, he selected. That's the first thing. And yeah, totally 7, right? Seed shape, seed color, pot shape, pot color, flower position, flower color, stem height. Okay, 7 characters. Okay, and contrasting characters. I told you contrasting character. What are contrasting character? See, listen, Hari. One is round means contrastingly it is wrinkled. Purple or violet means contrastingly it is white. Height, tall means dwarf or short. Okay, like that 7 contrasting characters he selected. And he crossed both of the characters. Both 2 plants he selected. One is tall. And here one term is there. He took plants with different character. Okay, can you see? He took plants with different characters, a tall plant and a short plant, crossed them, okay, produced a progeny from them and calculate the percentage of tall and short progeny. What it says? He selected plant with contrasting character. He crossed 2 contrasting character plant and calculated the progeny for their characters. One is dominant, tall is dominant, another one is recessive. Okay, fine. So here in the first, he selected the first generation. What is first generation? Or F1 progeny. All were tall. Look at it, first generation, all were tall. And then he selected all F1 generation. And then he got, means not all plant are tall. In the F2 generation, 1/4 of them are short. Means 3% tall and 1% short. Okay, and then he said, like, depending upon the percentage which he got, and he assumed, okay, how the characters are inherited. Okay, fine, don't worry. So this a general overview. What we are going to do is, what I'm going to do is, I'm going to work out. Do you all know how to work out monohybrid cross? Monohybrid cross, I'm going to work out how Graham Mendel did this experiment. Okay, that we are going to work out. Please listen. Come on, just a minute. Can you all able to see? Okay, mono hybrid cross. Okay, what we are going to work out? Mono hybrid cross. See, in most of your exams, you get this question. Okay, practice sessions, you get this question. How to do this? You can copy P generation, parent generation. One plant is, I'm just writing the genotypic character only. One tall homozygous and then one short homozygous. Okay. And second step is gamete formation. gametes. Like this only, the gametes will form. If you look at me, then only you can understand. Ari, can you able to do it? So the gametes are segregated now. Okay. And the F1 progeny forms. Okay. Copied till this yourself, Hari. So what you can understand from this? Listen, he selected two parents, P generation. This is monohybrid cross. What is monohybrid cross? Monohybrid cross. One pair of cross between one pair of contrasting character. Okay, look at that. This symbol represents male plant. This symbol represents female plant. So how do this is the genetic description TT, but look at that. What is this? This represents what? Homozygous dominant tall plant. Homozygous dominant tall plant. Another one is it is crossed with homozygous recessive dwarf plant. Okay, fine. Why it is homozygous, Hari? Because both the alleles are of same type. Homo, similar type of alleles. Capital T, capital T. Here small t, small t. T is dominant. Capital T is dominant. Another one is recessive. Okay, two parents he selected, and now these parents will produce gametes. Okay, what are the gametes? Look at that. This T separated here. Okay, this TT separated like this, and this TT separated like this. Okay, and then now F1 progeny. From that, see the F1 progeny are formed when these two parents are crossed. The progeny will have genetic genotype like this only. See, this is just to understand. This is what? Just to understand. Okay, the gamete will have either this gamete, this T or this T, this T or this T. Okay, now the two parents are crossed. You can see here this and this. What is produced there? Means both the character, both the alleles are there. See, this is genotype. What is genotype? The genetic composition, TT, that is genotype. What is phenotype? Phenotype. The visible trait are called phenotype. Okay. What is the phenotypic trait here? Look at that. Heterozygous. Why it is heterozygous? The plant looks tall only. Okay, the plant looks tall. Dissimilar alleles. Okay, one is tall and one is different alleles. Okay, short. But since the tall gene capital T is present, and you can see the plants as tall plant only. But what you cannot see here? Dwarf plant. When a tall plant for homozygous is crossed with dwarf plant for homozygous, how the progeny will be there? All will be tall. Okay, in this condition, all will be tall. Okay, fine. Now, the second step is crossing of F1 progeny. Okay, before that, the gametes forms. How the gametes forms here? Okay, how the gametes forms? Okay, how the gametes forms? This one, this one, this one, this one. Means T, T, and then T, and then T. Can you understand? The gametes are separated. Okay, these are all the gametes. Okay. Okay. Now we are going to work out for F2 progeny. F2 progeny. We are going to predict F2 progeny. Okay. So for what we use actually? Punnett square. Okay. What is Punnett square? Tell me what is Punnett square? Okay. And look at that. Put the gametes here. One is T, another one is T. And here T, another one is T. Okay, fine. Okay, now we are going to work this T, and what will come here? And here this T and small t. And here, why? See, always dominant should be first, written first, and then this T. Okay, and then what comes here? Okay, now you have worked out the Punnett square. Now you have to make a table or genotype ratio, genotypes. What are the genotypes present? Look at that. First one is T, T. Okay. And second one is, how many TT are present? How many are present TT? One. Correct? Only one. So write the one here. And second one. Okay. How many are present? Two. Strike that. So two. 1:2. 2:. இப்ப this genotype. Can you understand? So what is the genotypic genotypes? TT, TT, TT. What? How do you describe this, Hari? How do you describe this? TT, homozygous, dominant, tall. Second one, how do you describe this? Second one, heterozygous, tall. Heterozygous for tall. Second one is this one, heterozygous for tall. And here, dwarf. Okay, yeah, fine. Now you have to write the phenotype. Genotypes you have written. Now the phenotypes. How many were tall? This also will be tall. Okay. How many are tall? 1. Here also in heterozygous also it's look like tall only. So total how many are there tall? 3. Is there how many is dwarf? Tall 3, dwarf 1. Dwarf is 1. So 3:1. That is 3 tall, 1 dwarf. So 3:1 ratio is what? The phenotypic ratio. Can you understand? Is it clear? Can you all able to work out this? Okay. Is it easy for you to work out? On your own? Okay. Now I'll give you one task. Yeah, please copy. From where you have to start here? Here. From here, right? Phenotype. Just a minute. Okay. How many are tall? This also looks tall. Okay. Tall plant. 1 tall plant. And here, these two also, even though heterozygous, phenotypically how it looks? Tall. And this looks dwarf. Apo, in total, 2 plus 1, 3. 3 looks tall and 1 looks dwarf. So 3:1 ratio. Okay, fine. Just show me after writing, after copying, show me your book. You copied, huh? Come. Mono, speech generation. Okay. Even I too didn't write, but here write selfing of F1. Yeah, selfing. This is F1, right? F1 protein. Selfing of F1 means T, T into T, T. Okay, yeah? Good. 3:1 ratio. Good. P generation. Okay, I usually write it like G H. Actually, a P generation it is. Okay, generation male homozygous GOUS. Okay, dominant tall. Okay, into homozygous recessive dwarf. F1 progeny. Okay. And then here selfing off. See, I too missed there one thing. Okay, here you have to write selfing of F1. That is TT into TT. Okay, then only the gamete formation here. Okay, what the how the gametes will form here. T T T T, and these gametes only you are writing here. F2 progeny, and you should write here 100, because who discovered this? Reginald Punnett. That is his name. This square is called what? Prediction of genotypic and phenotypic outcome. Can you see? So genotypes TT, strike it, and then genotype TT, and then T. How many are there? 1. How many? 2. And how many? 1. So 1:2:1 is the genotypic ratio. Phenotypes tall, tall. So 2 together, what is this? 1 plus 2 equal to 3. Okay. And then dwarf. 3:1. 3 tall, 1 dwarf. Can you understand? So this is the monohybrid task. Now I'm going to give you a task. Okay. Honestly, you do it. Don't click it. Yeah. Okay. You can start working. Okay. Means another character. So let him come. Okay. So your task is right, work out the monohybrid for the following traits. Predict the genotypic and phenotypic outcomes of F2 progeny. Okay. Or versus okay. Hari, this is your question, Hari. Next page you go. Work out the monohybrid cross for the following traits. Predict the genotypic and phenotypic outcome of F2 progeny. What you have done for TT, small t, this you have to do it for round seeds and wrinkled seeds. Round dominant, wrinkled recessive. Homozygous round dominant and homozygous wrinkled recessive. Same. Completely what you have done starting from P generation, everything you have to do completely full. Next person, next page. No, you can now just look how it is done, and then on your own you have to do it without referring. First, just look into that, what it is that, what was done, and then you do it. Capital or capital? Round seeds. Homozygous, dominant, round. Another one is homozygous, recessive, wrinkled. Round na, the seed looks like this. Or means homozygous, dominant, round. Okay, round. And another one is or means homozygous, recessive, wrinkled. Okay, it's everything what I have written there, everything you have to do. Okay, round seed, wrinkled seeds. Do it. Hmm? Progeny. Progeny means offspring. to me. Anybody is there online? Kaushik? No, right? Okay, okay. But anyway, nobody is there. Participants also. Oh, audio is not connected. Huh? Dial in. No only. Okay. But oh, okay. I think audio may be recorded there. That once. Okay. See, where is the title? So now we should how to get rid of this audio. Correct. Okay. See, mono, hybrid, cross. Write the title. Okay. P generation. This is male and this is female. Okay. The gamete formation. Why you didn't write the gametes? Okay. Or. This is what? F1 progeny. Okay. You have to write this. And then F2 progeny. Punnett. Punnett. Punnett table or Punnett square. How the gametes will form? F1 progeny or the gametes, how do they form? Gametes. Or, 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 or. And what are the characters here? You have to write. Hetero, look at it. Heterozygous, round. All this has to be written. And then, okay, genotype. Or, 1, round, round, wrinkled. Okay. And then phenotypes. Okay. Round 3, wrinkled 1. Okay, good. Okay. So the little things you have to be very careful. Good. Give me your pen. So where is the title? Mono, hybrid, hybrid. Okay, cross. P generation. Okay. Or this is male and this is female. Or F1 progeny. Okay, and then gametes. Okay, gamete formation. You forget, then you managed it. Okay, heterozygous. What is this? Heterozygous star. Hmm? And then F2 progeny, Punnett. Punnett. Punnett square. Okay, selfing of F1. Here you have to write the gametes like this. Okay, the gametes. This one. Okay, and then right. Yes. S. Okay, RR1 you have written, RR2 you have written, and then RR1. Okay, this is white tall, white dwarf. This is what? Round. Okay, so like this you have to do. Okay, good. If you do it only, it looks simple, but when you do it only, you know what are the little mistakes you have done. Okay, fine. Okay, fine. Now, what you have to understand from this experiment? You tell me. From the monohybrid cross, what should be understood? Okay, so Graham Mendel concluded two laws. Okay, so listen, based on the monohybrid cross, Graham Mendel derived with concluded with two laws. Okay, what are the laws? One is called the law of dominance. Okay, another one is called what? The law of segregation. Okay, what are the two laws? The law of dominance and the law of segregation. Okay, law of dominance and law of segregation. Okay, fine. Listen to me first. What the law of dominance says. Listen, we are diploid organism. Ploidy means what? Chromosome number. Ploidy represents chromosome number. What is the chromosome number in our body? 23. N equal to 23. But we are diploid organism. Diploidy. So 2N. Ploidy means chromosome number. Our chromosome number is 23. N equal to 23. Okay. Okay. So ploidy means what? Ploidy means what? Chromosome number. Symbol is N. Okay. In humans, N equal to 23. But diploidy, we are diploid organism. So 2N. Upper 2 into 23 equal to what? 46. Okay, we have 46 chromosome in every cell except sperm cell and egg cell. All other cells we have 46 chromosomes. 7 days. After the production, sperm cell lifespan is 7 days in humans. If it is in fluid, see, if it is kept outside in the sense, if you chuma keep it here, it will die soon. Okay, no nutrients, no gas exchange, it will die. In male body, it can survive 7 days. Even if it enters into female body, it can survive 4 days. Okay, so total lifespan is 7 days actually. Okay, if it is frozen, freeze dried, 20 years also sperm cells can be preserved. How many years? 20 years. Can you understand? Depends upon how you store. But Excel storage is very difficult. Okay, fine. So chromosome number. So why I am telling this? What is the significance of this in the sense? See, we have many chromosomes, right? This is the basic structure of a chromosome. Okay. This is what? Centromere. This is what? Centromere. And there are two arms. Okay, you can see two arms. There are 4 types of chromosomes basically. Sometimes they have 2 equal arms. Sometimes they have what? Like this. One short arm, one long arm. And third one is actually one very short arm, one very long arm. Okay, third one. Okay, and fourth one, you can't even see the short arm. Like this only you can see. Okay, this chromosome are called what? Metacentric. This chromosome are what? Metacentric. The second chromosomes are called what? Submetacentric. Submetacentric. The third one is called what? Acrocentric. Fourth one is called what? Telocentric. What are the 4 types of chromosome? Metacentric, submetacentric, acrocentric, and telocentric. Okay, there are 4 types of chromosomes. Among the 46, we have 4 types of chromosomes. See, mostly you can see the first 3 types. In humans, what type of chromosome is not able to distinguish there? Telocentric chromosomes பார்க்க முடியாது humans-ல. Okay, it is present. All other 3 models only are present. Acrocentric, ACRO, acrocentric. Very, very short arm, very, very large arm. Okay, one part is very, very short, another part is very, very long. Okay, fine. And another term you should know is something called what? Homologous chromosome, non-homologous chromosome. Okay, what is homologous chromosome? See, look at that. Okay, chromosome number 1. This is chromosome number 1. This is chromosome number 1, which is what? Have you heard about this term? Paternal, maternal chromosome. Paternal in the sense what? Father's chromosome. Maternal in the sense what? Mother's chromosome. Okay, paternal and maternal chromosome. See, homologous chromosomes in the sense same size, shape, okay, size, shape, and gene locus. What is it? Gene locus means location of a gene. For example, here one gene is there. Okay, this is what? T. And here another gene is there. Can you see? When we write actually T, it represents what? In one chromosome, it is capital T. In another chromosome, it is location, same. Here, one gene is there. Here, the alternative form of a gene is there. 2 alleles occupies the same location on 2 different chromosomes. Can you understand? Okay, fine. Law of dominance. We are talking about law of dominance. Okay, listen first. Locus, gene locus, location of a gene on a chromosome. Gene locus means what? Location of a gene on chromosome. location of gene on chromosome. Location of gene on chromosome. Okay, location of gene on chromosome. See, look at this. Both of the chromosomes you are comparing. Both of the, what are homologous chromosomes? Homologous chromosome. Okay, they have same size, same shape, same gene locus. They are called homologous. Upper chromosome number 1. No, we know that, you know that chromosomes exist in pairs. Okay, chromosome exists in pairs. Okay, so same size, same shape, same gene locus. If it is, this is what condition? Homozygous or heterozygous? Homozygous or heterozygous? It is heterozygous. Heterozygous for tall. For example, if it is Mendel's pea plant, like this. Can you understand? So, what are the two laws? Let me simplify. Law of dominance is, in a diploid organism, for every trait there are two genes present. Okay, one from Appa we get. This is from paternal Appa. This is from Amma we get. Can you understand? For every trait we get. Every trait means what? Character. For every character we get two genes from our parents. One may be dominant, another may be what? Recessive. Okay, what is dominant? The character which is expressed in the F1 generation is dominant. The character which is expressed only in the absence of dominant is recessive. Okay, you should know, read that actually. Dominant and recessive, that's the first thing. Okay, fine. Law of dominance. I will simplify, but we will be revising these things again, but keep this in mind. Law of dominance. Characters are determined by factors. Factors exist in pairs. One is dominant, one is recessive. The one expressed in the F1 generation is dominant. The one which is expressed in the F2 generation is recessive. Okay, first thing. Another thing, both the law of dominance, it may be law of dominance, look at that. Second is law of segregation. See, here, this is the first thing. Gametes are there. F1 progeny, correct? In F1 progeny, how many dominant genes are there and how many recessive genes are there? In what ratio? 1:1 ratio, correct? 2 capital T, 2 small t. And look at that in F2. This is F2 progeny. How many dominant, how many recessive? How many T? How many T? Anyway, 3 are tall, but look at the gametes, genes. 4 capital T you can see and 4 small t you can see. Yeah, you just check. No, capital T, just check. 1, 2, 3, 4. And small t you just check. 1, 2, and 3, 4. Means, though it is dominant gene or recessive gene, they have, look at that, they segregate equally. Can you understand? And inherited to the next generation. It may be F1. Look at that F1. They are segregated. Dominant gene segregated. Recessive gene segregated. Dominant gene entered into the another generation. Recessive gene also entered into the another generation. Can you understand? So, in segregation and inheritance, there is no difference between a dominant gene and recessive gene. Only difference shown in expression. Phenotypically, they show differences, but genotypically, equally they inherit, segregate in meiosis and inherited together. Why meiosis I used? Gametes are formed by what process? Meiosis process. Can you understand? So segregation, that is law of segregation. Okay, in monohybrid cross, these two laws are derived. Okay, in tomorrow's class, we will be discussing about, next week's class, we will be discussing about dihybrid cross and another law of independent assortment. So before that, so please go through the yellow book material and your textbook and come. So which helps you. Okay, thank you. Take care.